Hi, I'm Jan Witkowski at Cold Spring Harbor Laboratory. This is now the third day of the 84th Cold Spring Harbor Symposium on Quantitative Biology, and the topic this year is RNA control and regulation. And I'm delighted to have Susan Gasser here from the Friedrich Miescher Institute in Basel. Susan, I did miss your talk last night, so perhaps we could begin with you giving me a synopsis of what you were talking about.
Happy to do that. So it was an unusual talk, both for me, I think, and the audience. So we've been studying the mechanisms of heterochromatin repression, so how different ways to keep genes silent. And most of that is at the level of transcription. So histone H3K9, the formation of heterochromatin. And we found in a screen 3 subunits of an RNA controlling complex came out in a screen for de-repression of heterochromatin. So this was in C. elegans, but this complex is conserved from basically yeast, even bacteria to humans. It's called the LSM complex, so like SM protein complexes. So we decided to pursue it, and we asked the question. All the other hits in this screen were chromatin modulators. So this is what we expected, right? So this was completely unexpected. We had to read up on, what is LSM complex. And what it is, it comes in 2 flavors. There's one form in the cytoplasm, one form in the nucleus. They share 6 subunits, and the 7th is either subunit 1 or 8, is either specific for cytoplasm or nucleus. So first thing we did was realize that it was the nuclear complex. That's good because it said that it was working at the level of probably of the genes. And we then looked at the proteins that are supposedly interacting with this. Its normal role is to bind U6 RNA, catalyze or sort of chaperone splicing, and then help trigger the degradation of the spliced out intron. So it binds an RNA endonuclease called, or exonuclease called XRN2. So we checked the proteins that are associated with it, and sure enough, they were also involved in this heterochromatic silencing. This was all done with a reporter. Then we asked the question, which endogenous genes are sensitive to this RNA degradation mode of silencing? And we detected, several hundred genes that were de-repressed, so upregulated in the absence of this LSM complex plus the XRN2. And we asked, what's the nature of these genes? So sure enough, they were all very poorly expressed or low expressed genes. And second of all, we looked at the histone marks on them to see were they euchromatic or heterochromatic. And they were all, 95%, carried the histone mark, H3K27 methylation, which is the polycomb mark. Now, polycomb is known to silence genes. It's usually considered to create facultative heterochromatin, so in a tissue or cell type specific manner. And it actually poises genes in an off state, but such that they can also be switched on in differentiation. So it's quite intriguing that all the genes that were controlled by this LSM 2 through 8 were facultative heterochromatin or polycomb marked. So we asked the question, how does it work? Polycomb normally represses by transcriptional repression, blocking the transcription. Right. But here we were working on an RNA degradation. Yeah. So we checked the genes that were sensitive to this RNA degrading complex, and showed that actually if you mutate the LSM8 subunit, which is the specific one, then sure enough you stabilize the RNA, the low-level RNA. So even though genes are repressed by polycomb, there's a low level of promiscuous transcription. And then we asked, were they the same genes that were regulated by XRN2, LSM8, and polycomb? And indeed, they overlap significantly, so they really are. And finally, we asked the question, does it feed back in any way? Does the LSM complex and the degradation of the RNA feed back to stabilize the transcriptional repression that's mediated by polycomb? And indeed it does. So we see that the K27 methylation mark is reduced. So what it constitutes is, you could say, a backup system, but a backup system that ensures the integrity of the polycomb repression pathway. So this is rather non-canonical. The polycomb field is not too keen on having something other than transcriptional repression through, preventing POL2. So it's been an interesting ride to try to publish this.
Right. So does LSM just mop up this low-level transcription, so to speak?
Right.
This breakthrough transcription.
It just breaks through transcription. So it degrades the messages. It makes usually they're full-length messages. At least they're not enriched in, let's say, just the N-termini of the genes. And there must be a decapping enzyme, and then the exonuclease XRN2 degrades it. So yeah, so it's basically reducing the level, often 10-fold, from the level that is expressed, which is, let's say, the background level of read-through of polycomb genes. It's known that polycomb isn't, 100% off. Heterochromatin, like centromeric heterochromatin, that can be more off.
Well, I was going to ask you, is this more general? I was going to ask, how, if a gene is repressed,
yes,
is it really repressed, or is there always a little bit of sort of?
Well, I would say it's quite repressed if you have both the transcriptional repression and the mopping up
yes.
of the promiscuous transcript. And I think when we knock out this secondary pathway, we're still seeing the animals are still alive. Their development is impaired, so this is C. elegans, and they become sterile. So if you ask how important is it, well, it probably is important. If the organism can't survive without it.
Yes, I would. Yes, I would agree with that.
Okay.
So, is this the first example of this sort of mopping up?
Actually, it's been observed and even characterized very well in pombe, fission yeast, and in plants. But it occurs at centromeric satellite repeats, and involves a different pathway because it involves a reverse transcriptase that makes a double-stranded RNA that's then diced, bound by an argonaut. It comes back and then nucleates the K9 methylation. So the H3K9 methylation, which is the canonical repression of constitutive heterochromatin. So the concept that a promiscuous transcript can help feed back and stabilize repression was already in the field, but it had never been demonstrated or characterized beyond fission yeast and plants. So this is the first example in animals.
And in animal, C. elegans is a funny little wormy thing. Is it also true of mice and man? Do you know?
Yeah, we've just initiated this, so of course the complex is conserved, and we find the LSM8, which is the unique subunit, on chromatin and associated with the XRN2. So, and we've just done the RNA-seq to see the genes that are misregulated. And what we see, we haven't actually fully characterized it, but there are genes that both go up and down, and now we're just trying to correlate if it correlates with the polycomb mark. So we're hopeful.
Another little thing about LSM, subunit 8 is
Yes.
the one that makes it nuclear, and
Yes.
1 makes it cytosolic.
Yes.
So, I mean, are these carrying some sort of signal that...
Yes. Well, so the complex as a whole recognizes message, poly A, in the cytoplasm, and in the nucleus, when it recognizes the U6 NERP, it recognizes poly U, small U stretch. So we don't really know if there is a signal on these polycomb repressed genes in the coding or the 3' end, or the somewhere, or the 3' UTR, that is actually recognized by this LSM2 to 8. But it's clear that, in a way, you could call this complex structural, because what it does is bring in an exonuclease. In both cases, in the cytoplasm, it also degrades messages, and in the nucleus, it degrades messages and introns. But the real link that's missing is what brings it to this class of genes. Why is it recognizing the polycomb genes? And so we don't know if it's a feature of the RNA. They could be aberrantly capped. They could be have some secondary structure or motif in the RNA. It doesn't seem to be 3' UTR specific, so we've swapped 3' UTRs. Anyway, there are hundreds of genes that are regulated this way, so it's not one single UTR motif. But we're looking for this signal.
You used the word non-canonical a bit earlier. That seems to be being used a lot at this meeting. I was thinking of Jeremy Willis's talk this morning, and RNA is clearly a much more interesting molecule than DNA.
Oh, I don't know, but the combination is fascinating, and I think there are many variations on DNA that we haven't actually explored. There are modifications, of course, methylation, but also in sequence motifs and sequence variation, and all the repetitive elements and how they behave. I'm still fascinated with DNA and chromatin, but RNA is fascinating and very non-canonical. I mean, I would say everything, the simple messages that we had about RNA, 50 years ago, are pretty much, water under the bridge.
Yes. We were talking just before we started about your institute.
Right.
We're commenting that this was founded by Novartis, or
Right.
by
Ciba-Geigy, 50 years ago. Actually, next year it's 50 years. And it's been funded by industry for 50 years. Initially, it was funded 100%, and the last 15, 20 years, about 70%. So we compete for competitive grants, which keeps us really linked to the academic world. And the fabulous thing is that for all that time, they've never told us what to do. So it's basic research, fundamental biomedical research funded by industry. First it was Ciba-Geigy and then Novartis. And they've been extremely generous. And fortunately, it's one of those instances where we can say that they recognize the value of open-ended research and the strength of curiosity-driven discovery.
That is rather rare these days amongst
Sadly.
companies, I think. Is the institute unique in these days?
No, there's the IMP, the Institute of Molecular
Yes.
Pathology in Vienna, is funded by Boehringer Ingelheim in a very similar manner.
Right.
And actually, Boehringer Ingelheim even has a second, smaller institute in Mainz, which is their headquarters. And I keep trying to convince companies that this is the way to go, that they have, a hotline into clever scientists, hopefully clever scientists, but just the energy of discovery-driven science is something that keeps a company on its toes, challenging the ideas that are sometimes become very inbred within drug discovery or drug development. It's not it's really drug development, and very often, yeah, they need to be shaken out of their ways of thought. So at least wisdom from the top has kept us alive, and hopefully for another 50 years.
Right. We can only hope that governments acted in the same fashion.
Right. Let's hope.
Thank you very much.
Thank you.